Thank you very much, David. So, I would like first to thank the organizer for inviting me today and to give me the opportunity to talk about Institut Pasteur Korea. As a nexus between basic science and clinical application. So the Institut Pasteur Korea, or IPK, was founded in 2004, and it's a result of a joint venture between Institut Pasteur in Paris and the Korean government. So Institut Pasteur in Paris was founded in 1887, and it's one of the oldest biomedical translational research institutes in the world. And the institute always try to link the bench to the bedside. And they combine excellence in basic science with applied research in the field of vaccine and diagnostic. And this business model has earned the institute 10 Nobel Prizes and the subsequent amount of revenues since they started a long time ago. So, what is the IPK mission? I think we are being created to accelerate the translation of basic research toward new medicine. So, and this is something not very obvious. It is called, what we refer to as the translational valley of death, where we have, on one side, knowledge providers like university, which are excellent in science, but they are really, quite also poor skill for translations. And on the other side, you have what you call innovation users, like pharma or biotech, which are excellent for clinical development and marketing, but they very often lack creativity. And IPK was set up to try to bridge these two worlds. So how do we do it for drug discovery? I think in the 2000, the model to find new drugs for new medicine has been influenced greatly by the genomic revolutions. And these revolutions led to a very reductionist approach in drug discovery. It was a bit like, Gene X give you this is Y, so you have a target. And for the target, you only have to do an assay and to screen and to find new medicine. But this very simple, rational approach has been usually disappointing. Because it's only at the end that you will validate your molecule that you found, and this might have taken maybe 10 years to find a molecule active in the target to be good enough to be tested in a disease model. And you might find out after all this time that this molecule you found, this new clinical medicine that you could have found, is not active in this model. It can be not active in this model, it can be inactive in vivo, it's very long and that didn't give any very good results in some field. So what IPK decided to do is to put a reverse approach, to put first the complex disease model. So what we try to do is to develop a very complex disease model in the lab, in a way, that we are going to use to screen compounds and find new leading medicine. So the advantage that you can, in this complex disease model, if you can do it, you can evaluate all the targets at the same time to a given phenotype. This is, of course, much faster to find biologically active lead molecules than the target-based approach. So how do we do it? So we develop some models which are complex and visual, and you use imaging and chemicals to probe gene functions and to find drugs. So, and I will show you an example with the Alzheimer's disease where I can show you that complexity can be quantified. So what we did for this project is to derive some neurons from patients which are Alzheimer's disease patients or normal patients. So what we do, we take fibroblasts from some people, do some induced pluripotent stem cells, from there derive into neurons. And these neurons are cultured in a theoretical plate format. And we can use confocal microscopy to observe in every well what is happening. And on these images, I can show you that the degree of complexity you can see in a well. This image, the macroscale, is what you see in a well. So you can see that the complexity of the neuron is quite huge. We can see many things. And as we zoom in at the meso and macro scale, we can see even more details. So, in this image, we can show that the neuron we can produce from the patients is very robust. This is different patients and different well. So each kind of patient gives different phenotype, but we can also quantify. We can also manipulate the branching and the morphology. If you do different coding, the neurons can give different morphology. And by developing some in-house algorithm, we can quantify very well all these phenotypes that you observe, and being able to see, this degree of complexity in this kind of disease. Just to tell you that this is a very good example of where we combined, different teams there, like biotechnology and IT, that allow us to really see things and quantify them very well. So, and when you're able to do this kind of images, we can have multiple applications, of course. We can use this model to screen for compounds that have an effect on neurons. We can use it for diagnostic. Can you find, can you predict which patients will have Alzheimer's later or other form of disease? It can be based on cell therapy, and of course, organic science is fantastic there, because we can maybe imagine, building some, mini brain there on the 3D, and we try to do it, and it's a very exciting project. So I can just to show you that complexity can be quantified very well by imaging technology. So now at that stage, I think I could show you that we can develop complex disease model that you can use for screening and find molecules that have an influence on this disease. So what you can do have a compound that give you a given phenotype, an effect, but what we don't know at that stage is how the compound does it. What is the target of this molecule, of this compound, because this needs to be found if you want to put a compound to the clinic. Because a cell is very complex, of course. So we have thousands of genes, and the compound can have an impact on any of these genes. And usually, the effect you see is only due to the one target. And this target has to be found to progress the project forward. So, and to find targets of any new medicine that we are trying to develop, we adapted a new technology, which is based on siRNA knockdown that we use for target identifications. And the idea is very simple. So what we do with a compound, with a drug, having an effect, can be done by target knockdown. If you use some small siRNA, there are small RNA that can knock down any genes in the cell, any of one after the other, we can knock it down. And if one of the siRNA block a target in part of our disease, we can see an effect, like for the compound. So how do we do it in the lab? This is the principle. So what we do everything in a glass slide, which are quite tiny. So the siRNA are printed on small slides from the robotic system. From there, we have the disease model that you culture on the top. We do some transfection, and after incubation, we can image again the phenotype and develop in-house some algorithm to quantify the phenotype. This is again, I think, a good example of integrated enterprise there, where you have some engineering, again, biotechnology and IT. So this is to give you an idea of the size of the slide that we use. On this slide there, we have 1/4 of the human genome. And this is compare this size to a classical plate that people use usually in the field. So, and even before we do things on this tiny slide, you can see again complexity. You can see phenotype. And being able to do this, approach on the glass slide is of course much faster and cheaper. So now how do we use these two technology, this approach to find drug target? Now we have a compound that give an effect, and we have this siRNA technology. So if you combine, both approach together, you can have a method to find the target of any given drug candidate that will impact on a given disease. So, and this is how we do it basically. So we have, it's the target of a compound. So you do some mRNA. So you add the small RNA, siRNA, that will reduce the pool of RNA. You have much less, meaning that we'll have much less target compared to untreated cells. And now, if you add your compound to this system, you have a shift in activity. The compound will be more active when you add the siRNA from without. And we can see a shift in activity if the compound, when the compound leaves the target. So basically, this is how we do it. We have a compound that gives an effect. And we have the genome on the glass slide, and every single gene is knocked down, one after the other. And we test in dose response the compound against every single knockdown possible in a cell. So basically, we have, we screen, very rapidly 24 genomes. And we use for that 120 slides for one project. If you compare to what you need, in a well-based, that corresponds to about 1,100 plates. So you can see that it's much faster and much cheaper than competitions there. So, and when we have done this, what we do, we analyze automatically all the response, with also algorithm. So we have here in the blue the dose response of the compound, and we look at, by automatically any siRNA that will shift activity of the compound. And like this, you can really quickly find the target of the compound. So, and I will just give you an example for the a project on the cancer, melanoma. So, we develop an assay where we can find compounds that induce senescence, a phenotype in a melanoma cell. So using this assay, we could find a compound called CMPDA that induces senescence, cell arrest in cancer cells. And we screen to find the target. We screen about 20,000 siRNA, and we could find one gene only that increased the potency of this compound. So you can see here, this just to show the data. So we have CMPDA compound at 18 nanomolar that got no phenotype. But if you put the compound same amount plus the target siRNA, we can have the phenotype. So using this technology, we can find target very, very quickly. And so what we did for this project. So now I want to show you how we, can help bridging, knowledge providers and farmers. So I'll give you two examples. So the first example is of tuberculosis. In this example, we bridge, INSERM in France, which is the French National Institute of Health in France, with Current, which is the Korean biotechnology. And in the second example, we bridge, Yon's University, local big university, with Sanofi Pasteur in the field of hepatitis B. So, on tuberculosis, we developed the first assay that allows us to screen for compounds that will block. What we did, we have an assay that can visualize migrating tuberculosis inside human macrophages. So here in red, you have the human macrophages, and in green, you can see the bacteria. So we developed an assay where you can quantify how many bacteria are inside human macrophages. And we can develop some assay to find compound that will kill these bacteria, of course. And all this technology is done in a P3 facility because TB is quite dangerous, as we talked about it before. So using this unique assay that only IPK can do, this is what we achieved. So we screened about 200,000 compounds in this project against tuberculosis in infected macrophages. And we could find a few compound series, and we focus on one. And after chemical optimizations and a lot of time, we could optimize this compound series to a molecule called Q203, which is the most active compound ever found against tuberculosis. This compound has a new mode of action, new target. It inhibits the cytokine DC1, Antibacterial. This is novel. So, if all go well, we expect the compound to be in clinical phase 1 in 2014. And if it is successful, this will be the first-class compound in Korea that reached phase 1 in humans. This has never happened in Korea as we speak. So, and this is, this program on tuberculosis is a very good example of creative economy. Because the compound, Q203 was licensed out to Korean Bioscience, which is an IPK spin-off company. And Korean, up to now, has been able to raise more than $10 million US from private investors. And it has currently 8 employees. So we get, we get financed by the Korean government, but we can create, value for IPK. So, for the program on hepatitis B, this is much early, so I don't have much to say there. Just that, the idea of the program is to find a cure for hepatitis B, which is a liver disease that impacts on many people worldwide, and there is no cure. Once you get HBV infections, you get it for life. So the idea of this project is to find a cure for HBV, which is based on the technology developed by Professor Liu at Yonsei University. This started maybe 1 year ago. So Sanofi invested in this project financially. And what's quite exciting that we'll have access to Sanofi compound collections to these programs. We can screen all the compounds from Sanofi against the assay developed by Professor Liu. So this is very new concept, but we are really excited about it. That you're able to bridge Yonsei and Sanofi, it's very important to us. So, so I hope that in this few slides I could, convince you that IPK in the last few years has been able to become an institute that can successfully translate basic knowledge into innovative therapy. And the guiding principle of the institute is to bridge, to be strong in, for now in the future in technology. Always, invest in technology to collaborate openly with pharma and with academia. We really are, highly collaborative institute. And in terms of team, the team is what makes the institute. I think we have a very, nice mix of people from all over the world, and very multidisciplinary. So by this I will stop here and be very happy to take any question.
Thank you very much.